Hi everyone, this is Lecture 9, Eukaryotic mRNA Processing. The material here is covered in Chapter 5 of your textbook, and really this lecture is an extension of the previous lectures we've had on transcription. We're focusing mainly here on what happens to eukaryotic RNAs after they are produced. To start with, let's look at a few quiz questions. Number 1, which of the following occur or occurs in the nucleus of a eukaryote? 2. What is an intron? And 3. What is the role of the poly A tail of eukaryotic mRNA? So eukaryotes and prokaryotes have some similarities and some differences. For example, the genes are organized differently in eukaryotes as opposed to prokaryotes. Prokaryotes often have genes arranged in operons, and so one long RNA is producing many proteins. Here we have, in eukaryotes, we have one gene, one protein per RNA. We have some similarities in that we both use a TATA sequence to initiate transcription via the promoter. However, the termination sequence is a little bit different. We don't have the hairpin or stem loop structure that bacteria have. We also have a much more complicated regulatory system. We have not just one RNA polymerase, but we have 3. RNA polymerase 1 primarily transcribes small nuclear RNAs, what we'll talk about in a little while. And also, RNA polymerase 1 transcribes ribosomal RNA. RNA polymerase 2 transcribes messenger RNA, primarily. And RNA polymerase 3 in eukaryotes primarily transcribes transfer RNA. And the focus of today's talk is mainly on what happens to those RNAs once they are transcribed. We're going to be focusing on 3 things. What happens at the beginning of messenger RNAs, what happens at the middle, and what happens at the end. Let's start in the middle first. Eukaryotic RNAs are produced in the nucleus, as all RNAs are. However, they consist of different regions, some of which make it into the mature messenger RNA and others that do not. The portion that is retained and eventually expressed in the messenger RNA are called exons, for expressed. Those are shown in yellow on this slide. Part of the RNA, though, is removed and does not show up in the mature messenger RNA. These are called introns for intervening sequences. There are specialized enzymes, which are depicted here in green, which remove the introns. The protein in eukaryotes then is produced from messenger RNAs that have been spliced. The introns that are removed, and only the information in the exons is encoded into the protein. The process of cutting out introns is rather complex. I just wanted to touch on a couple points here. The enzymes that cut out the introns are actually not protein enzymes, they are RNA enzymes. We call these ribozymes. In this picture, they're depicted as little balls, U1, U2, and all the way through U6. You notice that these balls are binding to the intron, which is shown in blue on this picture. These RNAs, called small nuclear RNAs, are collectively known as spliceosomes. So the entire structure that helps cut out introns is known as the spliceosome. These small RNAs, the small nuclear RNAs, combine with proteins to form what we call small nuclear ribonucleoproteins. It's kind of a mouthful, so we use the word snRNP, or better yet, SNRPs. SNRPs then are the combination of the RNAs plus the protein. In this picture again, they're depicted as U1 through U6. Notice that U1 binds to the 5' end of the intron, and U2 binds to the so-called branch point, where there is an A typically. U1 then is removed, and other SNRPs come in to fill its place. They combine with U2 to help catalyze the removal of that 5' end of the intron, followed by the removal of the 3' end of the intron. An interesting lariat structure is formed. That looped structure is formed before it's removed. So that's what happens in the middle of eukaryotic messenger RNAs. What happens at the beginning? A so-called cap is added to the 5' end. This cap is kind of an unusual nucleotide. It is a guanosine, however, it is added in the opposite or reverse direction. It is added via the 5' carbon and the 3 phosphates. It is not added by RNA polymerase. Remember, RNA polymerase has already transcribed the RNA by this point. This cap is added post-transcriptionally. That means after transcription has completed. There's a special capping enzyme that does this. This guanosine is modified at the 7 position in the guanosine to have a methyl group, CH3, so it's called 7-methylguanosine. That cap structure serves an important biological role for this RNA, which marks it as a messenger RNA and directs it to the ribosomes for translation. We'll see more about that in a second. So messenger RNAs in eukaryotes begin with a 3' methyl G, meaning that it's going from the 3' connected to the 5' connected to the 5' of the messenger RNA. In addition to helping it get to the ribosome, it protects the 5' end, identifies the RNA as a messenger RNA. And again, it provides a binding site for the ribosome. Now, at the end of a messenger RNA, there are special termination sequences. Again, they're not stem loop structures that you find in prokaryotes, though. In particular, one sequence in the gene AAT-AAA is transcribed to AAU-AAA, and that serves as a signal sequence to terminate the transcription. About 10 to 30 nucleotides away from that AAU-AAA is a C and an A. And about 30 nucleotides away from that CA is a GU-rich or a U-rich region. That is actually not unlike the bacterial situation where there is a U-rich region that helps destabilize the RNA-DNA hybrid. That RNA in this picture is color-coded blue and kind of a yellow-green color on purpose, because what happens is right after that CA, right after the blue segment, the remaining part of the messenger RNA, the RNA, is removed. It's cleaved off by specialized enzymes. Once that happens, then another enzyme has created a long stretch of A's called a poly-A tail, and another enzyme attaches that poly-A tail to the C and the A. So that the mature messenger RNA ends at that CA and is followed by a string of poly-A's. What's the point of having a poly-A tail? Well, proteins bind to this poly-A tail, and those proteins, which in this picture are depicted as these green circles, interact with specialized initiation factors called IFs, which bind to the cap. That way you have a cap protein bound to a poly-A tail protein. It serves to link the two together. It serves as a quality control step, showing that if we're going to translate this RNA, which is what these green ribosomes are doing, we want to make sure we have a proper 5' cap and that we have a proper poly-A tail. If we don't have one of those two things, translation will not begin. So it serves as a quality control to make sure that the RNA is truly a messenger RNA. Finally, just to show you how this all sort of fits into a cell, the DNA is, of course, inside the nucleus, which here is shown in this white circle. The DNA contains the genes, which are composed of exons in orange, dark orange, and introns in light orange. This DNA is transcribed by RNA polymerase into a primary RNA transcript. The exons here are shown in blue, and the introns are shown in yellow. Post-transcriptionally, so after RNA polymerase is done, the capping enzyme adds the cap, which is shown here in green. The introns are spliced out, and the poly-A tail is added. Once those three things are done, the messenger RNA is exported out of the nuclear pore, out of the nucleus, into the cytoplasm. It is then translated by ribosomes. And there you have it, messenger RNAs from eukaryotes. See you in class.